Bismillahirrahmanirrahim. Today we do the another paper of O-level biology, and this is a question paper double two, variant double two for November 2020. As you can see, this paper is for 1 hour and 45 minutes, and we need to be very careful about this time period so that we can do all the questions in the specified time. The first thing we do is we read the whole question and then we try to attempt the parts of the question. The flow chart shows stages in the life cycle of a flowering plant. So stage one is pollination. Pollination is the transfer of pollen from the anther to the stigma. Then the pollen tube formation, as you know, as the pollen lands on the stigma, then it starts to release enzymes, and a pollen tube grows down the style. And then the pollen nucleus is going to pass through the pollen tube and result in fertilization with the female gamete. Then there is going to be fruit and seed formation. You know, the ovule will become the seed and the ovary will become the fruit. So the ovary wall will be the fruit wall. And then the fruit and the seed has to be dispersed. Then step 5 has been left as an open-ended question. And then we have plant growth and flower formation. Now it says the name of the stage 5 of the life cycle has not been included in the flow chart. So we have to give you that, and that is during but then they give you a clue that during this stage a radical and a plumule are formed from a seed. So that has to be germination, it can't be anything else. Then it says state in the correct place on the flow chart the name of stage 5. So you had to write that here. This is stage 5. So you had to write here germination, and that would have got you the mark, the 1 mark, which is for this part of the question. So this is the 1 mark, and you would have got that 1 mark for that. Then it says describe the role of enzymes in the plant's life cycle. So that is now another for 3 marks, and we will have to give you the role of the enzymes in this stage of the plant's life cycle. This stage, so they're talking about this stage, stage 5, which is the germination. Now, as you can see, the role of enzymes in the process of germination, we've done it in great detail, is breaks down, you can say, or you can say digests, or you can say hydrolyzes. If I put a slash here, it means it can be either this or this word, but this will be 1 mark scheme point. Even if you wrote all 3 of them, it will only get you 1 mark. Then the enzymes which work, because you see seeds have cotyledons and seeds have food store in that, and that has the main 3 components of the cotyledons is starch, which is a carbohydrate, and proteins and fats. That's why you see when you crush seeds, you get oils out of them because they have fats in them as well. So it could have been amylase, could be maltase, could be lipase, could be any other enzyme like a protease. But that would only get you 1 mark if you named any of these enzymes. And if you named all of them, even you'd only get 1 mark. Then what does the amylase do? It converts the starch to maltose, maltose to glucose by the enzyme maltase. And finally, what we need this glucose molecule. Why? Because this glucose has to be respired to release energy. Then if we used it, protease will convert it into amino acids. The amino acids will be used to make new proteins in this growing radicle and the plumule and the little seedling growing out of the seed. Well, let's look at the B part of the question. It says 2 of the stages in the life cycle of the flowering plant may depend on the action of an animal. Identify each of these stages. For each stage, describe how the action of an animal may be required. Now, stage 1 was pollination. In a pollination, what do we need? We need an insect. So insect pollinated flowers. So they move, or the insects transfer the pollen. So that's the first mark scheme point from anther to stigma, and of different flowers. Like if you have a red rose bush and a white rose bush, so the pollen transferred from the red rose bush to the white rose bush. Then the second one was stage 4, in which you could have said fruit dispersal or seed dispersal. And what happens in this is that, as I explained to you, and even in my video, it says that, berries will be eaten by birds, and then the birds will fly far away from the parent plant and may go somewhere else, and then those seeds will be passed out undigested in the feces. So birds eat the berries and then fly away, and then the seeds can be deposited maybe miles and miles away from the parent plant. So number one, the animal eats or consumes and they're not digested. Then the animal moves, like birds will fly away, and then they will be passed out in the feces, or you can say egestion, or sometimes we can also talk about the seeds having spikes on them, and then they stick to the fur of the animal, and then the animal walks away, and then somewhere else it just sort of brushes itself and the seeds fall off. So you could have talked about that also, because that is also a form of animal dispersal of fruits and seeds. So these were the 2 points that we had to discuss in this question, and this was for 6 marks. So the naming it, you can see there are many points in each of the part. In this one, there was 1, 2, 3, 4. In this one again, there was 1, 2, 3, 4 points. So there were 8 points for a 6 mark question. And I'm sure many of you would have got all those 6 marks. So in question 2, it was a little long sort of a question, and you have to read it all. In 1931, a scientist was pouring the powdered form of a chemical called PTC into a bottle, and a small amount of the powder accidentally blew into the air, right? A short time later, another scientist working in the same room said that she had a bitter taste in her mouth from the powder in the air. So you can only taste something if it has reached your tongue somehow. Can't be through your nose. You can't smell it. It said taste. So that is what you have to really figure out. The taste word you have to figure out. Then it says describe with reference to named components of the nervous system the nervous pathway that led the scientists detecting that the powder tasted bitter. So what is it? What is it? This is an electrical story in which you had to tell me that there was an electrical impulse which was generated. Why? Because the receptors or the taste buds in the tongue or the sensory endings in the tongue were stimulated, and thus generated an electrical impulse. And it was carried by the sensory neuron, and this information went to the brain, and then the brain analyzed it and said, okay, well, this tastes very bitter. Like for instance, if you put something you haven't tasted it before and you put it in your mouth and you say, oh, this is very sweet, or this is very bitter, or this is very sour. So these are the different things that you can taste, and we have all these different taste sensory receptors in the tongue which can actually tell all these. So 4 mark scheme points for a 4 mark question, which you needed to understand was the whole story about the reflex action. And this is, of course, a part of the sensory neuron. Here, of course, there's no motor neuron involved, but of course, the marks are for an impulse, receptors, taste buds, sensory neuron, and the brain being involved in analyzing it. This pouring the powder did not detect any bitter taste. It was later found that inheritance of two forms of the same gene determine if a person is able to detect a bitter taste from PTC or not. State the term used for different forms of the same gene. Like we say, for blood group, there are 3 alleles, IA, IB, and IO. So allele was the answer. Then several years afterwards, it was found that environmental factors cause people to detect a range of different strengths of bitter taste from PTC. Now state the type of variation. So people can detect different strengths of the bitter taste of PTC. Now this would be, of course, it says variation. State the type of variation. Now variation is only of 2 types. One is continuous and one is discontinuous. In discontinuous, there are few, 2 or 3 or 4 types. But in continuous, there is a large variation, and there are many, many intermediate values. So this would be continuous variation. Now, if you look at the C part of the question, it says chemicals similar to PTC with a bitter taste are produced by some plants to prevent them from being eaten. Animals that eat only plants have fewer genes that enable them to detect chemicals that taste bitter than animals that eat both plants and animals. So state the trophic level of plants in a food web. Well, plant is always the producer. It is the first trophic level, or trophic level 1. It says either you said producer, or you said trophic level 1, or you said the first trophic level. Then state the term used for animals that eat only plants. Very obvious. Herbivore. State the term used for animals. They are herbivores, but you couldn't have said primary consumer. So that is what you could have made it wrong, is primary consumer was not allowed here. Then it says they suggest why animals that eat only plants have less or fewer genes. So herbivores have fewer genes that enable them to detect chemicals that taste bitter, and animals that eat both plants and animals have more genes. Now the reason is that all energy and all nutrients come from plants. We all producers. So herbivores, the animals that are herbivores will have less because they have less variety of plants to eat from. And food and plants taste less bitter. But the animals that feed on both plants and animals have a bigger variety of food, so they have more genetic information. And of course, in both these situations, it's the survival of the fittest. The herbivore has to be adopted for its role, and the carnivore has to be adopted for its role. Question 3. When an apple fruit is cut open, the cut surface of apple tissue quickly becomes brown and is less likely to be eaten. This change in color is shown in the diagram. White apple tissue immediately after being cut, and then the brown apple after a short time after being cut. So this is the information they've given you in the question. The change in the color of the apple tissue, the change in the color of the apple tissue is due to a series of chemical reactions. An enzyme called PPO. An enzyme called PPO acts as a catalyst for one of these reactions. State what is meant by the term catalyst. Did not say state what is meant by the term enzyme. State what is meant by the term catalyst. Now, as you can see, what is meant by the term catalyst is increases the reaction speed. They speed up reactions, and they are not changed up by the reaction, or they're not used up in the reaction, and they can be reused again and again. And of course, the third mark scheme point is decreases the activation energy. So three possible mark schemes for a 2-mark question is what is meant by the term catalyst. And part 2 of the question says the change in color can be prevented by placing the cut surface of the apple tissue in boiling water for a short time immediately after the fruit is cut. Now explain this observation using the lock and key hypothesis of an enzyme action. Well, it makes sense. It's an enzyme which brings about this change of the brown color. So if you place it in boiling water, of course it's going to denature the enzyme, and the enzyme will not be able to work, so it won't turn brown. So you had to explain this using the lock and key hypothesis. Possible answer to that was as soon as you put it in boiling water, the active site of the enzyme would be changed. And this, of course, name of this process called the enzyme denatured. So you could in one sentence get 3 marks. Like if you said the enzyme's active site loses its shape, enzyme is denatured, and the substrate does not fit anymore. So it's not the number of sentences that you write that you will be marked for. You'll be even in one sentence, you can get 3 marks. So, but basically, where would you go wrong? You would say the shape of the enzyme is lost. Well, that is wrong. Shape of the enzyme might remain the same. You see, this is an enzyme, and this is the active site. Now, the rest of the enzyme might change the shape. This enzyme might change shape here, but that doesn't matter. If the active site is the same, well, the active site is where the substrate fits. So you had to say the active site loses its shape. Active site loses its shape. And we say the enzyme is denatured. So there's a change in the shape of the active site, and this is called denaturation of the enzyme. And this substrate now can no longer fit. Like for instance, in this situation, this is the substrate. So there is a specific shape of the substrate which can only fit this active site. But if the active site shape is changed, if the active site now becomes like this, well, this substrate is not going to fit into it. And if the substrate doesn't fit, then no product is formed. So this is what you had to understand in this 4-mark question. What had you to give in as the answer? Now the part B of the question, which was for 3 marks, says scientists have recently used the process of genetic engineering to develop a type of apple that does not produce the enzyme PPO in its tissues. Suggest possible advantages of this type of apple. Now I always tell you, suggest is the one which is going to see whether you get an A star or not. It's the most difficult part of the paper. So if you know it, then do it. Otherwise, don't waste your time on it. I mean, as far as I'm concerned, you can come back to it and write 1 or 2 points which might be or might not be correct. But do not spend time on the suggest. The suggest is the most difficult part of the paper. So they make it purposely such a way that the paper is tough and the suggest part is the most difficult. So no browning, no color change, more likely to be eaten because they don't turn brown, so we won't waste them. Then increase sales or more profit, and we will no longer need to put them in boiling water. So we have 1, 2, 3, 4 marking scheme points, and you have to give me any three of these to get your 3 out of 3. Question 4. Hydroponics is a technique used to grow plants without soil. The diagram shows plants being grown using hydroponics. There's a light source, there's a nutrient solution supplied to plants, there's a plant, and there's the nutrient solution, there's a growing tray, there's a timer, there's an air pump, there's an air stone, and there's a nutrient solution pump. So please look at the diagram very carefully and see what they're trying to show you and give you. Then we come on to the question A. State the leaves of the plants are provided with a source of light for photosynthesis. State in either words or symbols the equation for photosynthesis. Now that's a very direct question. Everybody knows that, I'm sure, and you all can do that part of the question. So you give me the equation 6CO2, 6H2O, C6H12O6 gives you 6O2, and you got your 2 out of 2 in this part of the question. Part of the question said the roots of the plants are provided with a nutrient solution. The nutrient solution contains magnesium ions. Air is pumped through the nutrient solution using an air stone that contains many very small holes. Explain the advantage to cells the plant's roots are pumping air through the nutrient solution using the air stone. And the B part says state the type of cell and plant roots that absorb magnesium ions from the solution. State the process that transports magnesium ions against a concentration gradient into this type of the cell. More bubbles would mean more increased air passing to the roots, and more bubbles means more surface area of the air. Then what does air contain? It contains oxygen. Where is oxygen needed? Oxygen is needed for aerobic respiration. Aerobic respiration releases energy, my favorite word, releases energy. And what is this energy needed? This energy is needed for growth, and we need energy for active transport of ions. So there were a lot of marks for that, and there were 4 marks which you had to give me. So whenever you look at the marks, you must read the marks of each part, and then you must give me at least 1 or 2 extra points so that you are sure that you've got your 4 out of 4. Now look at here, we had so many mark scheme points: 1, 2, 3, 4, 5, 6, and 7. So 7 mark scheme points, and you have to give me any 4. So this is how you're going to learn to do these papers, is that you will write more so that you're sure you get your marks, the number of marks, and no marks are cut. State the type of cell in plant roots that absorb magnesium ions from the solution. Well, naturally it's a root hair cell. Hair was the word which was required. Then state the process that transports magnesium ions against a concentration gradient. Well, that had to be active transport, can't be anything else. Now, as you look at this part C of the question, it says farmers grew groups of 20 tomato plants for the same length of time in nutrient solutions with a range of different concentrations of magnesium ions. So when he says different concentrations, you have to understand could be 1%, 2%, 3%, 4%, and 5%. They determined the best concentration of magnesium ions for growth by measuring the following for each group of 20 tomato plants. Number one, the mean height of the tomato plants and the mean mass of the tomato plant stems and leaves. So the results of the investigations are shown in the table. And here you can see concentration of magnesium ions in parts per million. And then you have mean height of tomato plants and stems in centimeters. And then you have the mean mass, and this is in grams, of tomato plant stems and leaves. So we have 0 magnesium, 20, 6.3, 50, 30.3, 11.7, 32.6, 12.3, 23.4, 10.4, and so on. But then you see there is some sort of linkage here. Look, it's increasing up to here, and then in this one it is decreasing. Similarly here also, if you look at it, it's increasing up till here, and then it's decreasing. Then it says state the best concentration of magnesium ions for growth. And that, of course, would be 125. Why? Because look at this, the highest. Mean height was the highest and the mean mass was the highest. So that is why 125 would be the one which was had the best concentration magnesium ions for growth. And then it says explain your answer using the results of the investigation and your knowledge of importance of magnesium ions to plants. So now the first one was largest height and mass. Magnesium is needed for the chlorophyll molecule, just like iron is needed for the hemoglobin molecule. Then more photosynthesis, more chlorophyll means more photosynthesis and more production of glucose, or if you said starch or carbohydrate or sugar. So any of these was allowed, but I think production of glucose was the best to say, because then of course if it's in excess, it's going to be stored as starch. So production of glucose. And that was a 13-mark question. The diagram shows the total volume of water entering and leaving the organs of the human alimentary canal during one day. So let's just now circle these. 3,500 cm cube added by food, drink, and saliva, 1,500 cm cube added by the stomach, 4,000 cm cube added by the pancreas, and 200 cm cube added by the large intestine. So you can see I've added the 3,500, 1,500, 4,000, and 200, and you get a figure of 9,200. While here I've added the 1250, as you can see in this question, the 1250 and the 150. This is the one which I've added here, and this is the one which I've added here, and you get a total sum of 1,400. Now 9,200 minus 1,400 is 7,000. Basically it is 7,800, right? Now this is what I've done the working here. Now we proceed. The organs labeled A and B, and that of course was the esophagus and the stomach. Calculate the volume. This was 7,800. So you got a mark for that. To write it where the space was given to you, and you needed to understand the working, but it did show you how to show the working, so it's okay if you just wrote down the answer, and that was all that was required. Part 3 of the question says describe the absorption of water from the ileum, and that was osmosis, or you could have said diffusion. If you said osmosis, then down the water potential gradient. If you said diffusion, then down the concentration gradient. Then where did this happen? In the villi and the microvilli in the small intestine. So that was where it happened. Then where did it go into? It went into the blood or into the plasma, if you said, or if you said into the capillaries, even that was allowed. Then it says state how a named organ causes some of the water absorbed from the ileum to later become water in the urine. Like if it's a cold day and you drink a lot of water or you have a lot of fruit, then of course you'll be going to the toilet very often because you'll be urinating more often. And what is going to happen is that in the kidney, which is going to remove the water from the blood. So basically all the water that you eat, either in the form of fruit or fruit juices or just plain water that enters the blood. So the kidney will remove this excess water from the blood, and then it's going to be passed out as urine. The essay questions in the diagram shows a vertical section through human skin in a hot environment. Name and outline the role of P and Q in the control of body temperature when a person is in a very hot environment. So P and Q. So P is the sweat gland. And Q could be sort of the blood vessels, and well, you could talk of the capillaries. So blood vessels which are bringing blood to the skin capillaries, to the skin arterioles or the skin capillaries, where more blood is either going to flow through them or less blood is going to flow through them. Working scheme for this was number 1, P was the sweat gland. Produce sweat, sweat evaporates, and this removes the heat of the body, so cools the body down. Q was a capillary, and in this, of course, we are saying there's going to be increased blood flow, because if it's a very hot environment, there has to be vasodilation. So more blood flowing in the skin arteries, so more in the skin arterioles and more in the capillaries. And this blood is near the surface of the skin, so this is going to remove the heat from the body very readily, and the body is going to cool down. So this removal of heat, you either were going to get 1 mark of writing it here, or you're going to get, if you wrote it in any both the places, you could only get 1 mark for it. So you could only get once a mark for a certain point. Now explain how the process of temperature control in humans is an example of homeostasis and negative feedback. In homeostasis, we maintain a constant internal environment. The temperature of the body is maintained, so the enzymes are not denatured. Because otherwise, if the temperature fluctuates of the human body, then enzymes would either go in fast motion or will slow down and will not be able to work. So the body will go into slow motion. In negative feedback, there is a change. The change can be either an increase or a decrease. Like for instance, we enter this room is very hot. So for a short while, your body temperature and your blood temperature is going to increase. And then something happens and there's going to be a correction or a reversal back to normal. Like for instance, you eat something sweet, your blood glucose level rises, but then insulin is released and the blood glucose returns to normal. The kidneys are organs of excretion. Define the term excretion: removal from the body of toxic metabolic waste. I mean, that's the definition that we remember from the syllabus. But the toxic metabolic waste is what? Urea and carbon dioxide. So this was a 3 mark, and there's a very narrow marking scheme. You had to say removal from the body of toxins or toxic metabolic wastes, and the metabolic wastes are urea and carbon dioxide. A person with kidney damage requires the process of dialysis to take place several times each week. Dialysis is carried out using a kidney machine connected to the blood supply of the person. The diagram shows a section through the internal structure of a kidney machine. Blood to person, dialysis fluid in and dialysis fluid out. Blood from the person goes in here. So you see, you have to understand this, and then this blood is going to go back to the person here, and you can see this has been underlined is here. And the dialysis fluid, which is, let me give it another color, dialysis fluid is going to flow out here, and the dialysis fluid is going to flow in. It's sort of against the current. If the blood is entering from one side, the dialysis fluid is entering from the other side. Now, the structure of the dialysis membrane and how it functions is because it's very coiled and long and has multiple tubes. Then it has a larger surface area, then it's partially permeable and through which diffusion and osmosis can take place. And urea and ions can water can pass out, and large proteins and blood do not pass out. Basically, the dialysis is to remove the urea from the blood of the person whose kidneys are not functioning. Now, why must the dialysis fluid be continuously replaced is so that it maintains the gradient? Because the urea is passing out into the dialysis fluid. Now that must be removed. In must be coming the dialysis fluid with zero urea in it, so that the blood urea must come out. And this will prevent the buildup of urea. And this maximizes diffusion. So on one side you have the patient's blood, and on the other side you have the dialysis fluid. Now what is happening? Urea, so let's give it another color. Urea is passing into the dialysis fluid. This dialysis fluid must be changed because after some time then this will also contain urea. The blood also contains urea. There will be zero diffusion. So maximizes diffusion, maintains gradient, prevents the buildup of urea. So here is the human blood and here we have urea in it. Urea must enter the dialysis fluid and then it must be removed. So the dialysis fluid must be changed continuously so as to maintain the gradient and so as to remove, so that we can save this person whose kidneys have failed and he can be on the dialysis machine sometimes even for many, many years. And that finishes this paper, and we will continue with the other paper in the next video.